Differentiation of B cell into the plasma cells. Now, how does this differentiation of B cell in the plasma cell take place? The immunoglobulins that are synthesized by the B cells are inserted in their cell membrane, and the B cell cannot secrete immunoglobulins into the plasma. These immunoglobulins display on the surface of B cells are known as the antigen receptors. And the B cell displaying their antigen receptors, with the diagram already shown that the antigen receptor inserted into the membrane of B cell is the IgM. This heavy chain domain of the membrane bound IgM is associated with the two transmembrane protein. These are the Ig alpha and the Ig beta. This cytoplasmic portion of the Ig alpha and Ig beta possesses the immunoreceptor tyrosine based activation motifs, which is called the ITAM. This ITAM is made of the two precisely spaced tyrosine residues that are 9 to 11 amino acid residue apart. When an antigen enters the body, first epitope binds a complementary antigen receptor. The antigen bind to that receptors, then the antigen binding cause the oligomerization of the neighboring IgM molecules. After this, the oligomerization activates an intracellular tyrosine kinase, then that is FYN, and some other cytosolic tyrosine kinases such as LY and BLK and may also be activated. And these activation of the intercellular tyrosine kinase for phosphorylates in the ITAM of the Ig alpha and the Ig beta, and these phosphorylated items acts as a docking site for a protein, that is SYK. This SYK, which is also a tyrosine kinase, is activated, and this activated SYK phosphorylates the tyrosine residue of the some other mm, target proteins. Here we can see with the diagram when the antigen is interacting, these are the phosphorylation process is taking place, these are ITAMs and there is the SYK, these all are interacting with each other, and the Ig alpha and Ig beta can be seen here, and these are these how this interaction is taking place. Then the phosphorylated target protein generates some signals, and these signals then stimulate the B cells to differentiate into the clones of the plasma cells. And these plasma cells then start to synthesize the IgM of the different kind. And these IgM can be of the two types, membrane bound form and the secreted form. The two forms are synthesized from the same hnRNA by alternative splicing. And this splicing which occur in the B cell produce the membrane bound form, and the splicing which occur in the plasma cell produce the secreted form.